Now the fifth lecture deals with, or it will be dealing with, I will be talking about DNA replication. So let us start with it. Now replication. Now what is replication? Replication means replication. The word itself signifies, it is self-explanatory. It producing replicas, similar copies. That is replication. So here we are talking about DNA replication. Now DNA replication, for the first time, it was studied and elaborately described by Watson and Crick in 1953, along with the discovery of DNA. That is, during the discovery of DNA, when they proposed the model of DNA, along that time, even the replication process of DNA was explained. Now, replication of DNA is said to be semi-conservative. Semi-conservative means that the parental DNA does not break down completely, but 50% of it is present in the daughter DNA that will be produced from the parental DNA. So, replication process will obviously increase the number of new copies. So the new copies that will be formed, the new DNA that will be formed from the parental DNA will have, each of them will have 50% of the parental DNA, and hence DNA replication is referred to as semi-conservative. The parental strand, that is the original DNA from which the daughter DNAs will be produced, is referred to as the parental strand or the template strand. And the other one is referred to as the complementary strand. So one of them will be parental strand and the other will be the complementary strand. Now new DNA, one parental strand and one newly synthesized strand, as already mentioned, and hence it is called as semi-conservative. Now some, again like the previous time, always some proof is required. Proof to substantiate that yes, DNA replicates in a semi-conservative manner. Some procedures were carried out, some steps were carried out by Masselson and Stahl. Again, the experimental material here is was Escherichia coli. Matthew Masselson and Franklin Stahl in 1958 carried this experiment to prove that DNA replication is semi-conservative. What they did, they took medium. Now what is a medium? Medium means a solution which may be semi-solid, which may be converted to semi-solid, or it may be kept as a solution form containing all the essential elements or minerals required for the growth of any organism. We want to grow the organism artificially in the lab, then we have to provide it the food material. So the food material will be provided through the medium. We prepare the medium, adding all the components required by the organism in definite proportions, add the organism into that medium and let it propagate, let it grow. Such a solution is known as medium. Now, for this particular experimental proof, the medium was prepared with all the components that a particular bacteria, especially Escherichia coli, will need for its growth. In that medium, we have to add chemicals as sources of different elements. So, the source for nitrogen, ammonium chloride was added. Ammonium chloride was added into it. Now, the two types of such media were prepared. In one of them, the source of nitrogen, ammonium chloride, was radio-labeled using the isotopic form of nitrogen N15. Whereas the other media, the same with the same components, even in it the nitrogen source was ammonium chloride, but in it the nitrogen taken to prepare the ammonium chloride was the naturally occurring nitrogen N14. Now the bacteria taken was Escherichia coli. Escherichia coli, for a record, we have to remember Escherichia coli completes one generation in just 20 minutes. So in a day, it can complete many generations. Then after allowing Escherichia coli to grow in this media, they were centrifuged. They were subjected to very high speed rotation in centrifuge machines inside centrifuge tubes, and the solution taken in during the process of centrifugation was cesium chloride. These are some of the points that we need to remember. So the solution used was cesium chloride during centrifugation, and after centrifugation, according to the density, the cells they separated, which we will be seeing in the next slides. Similar experiments were also conducted in plants, that is Vinkia feba, by Taylor in 1958. Same year, they also carried, and the conclusions were similar. For the procedure to start, what was done? First, the solution, the media was taken, which contained ammonium chloride with the nitrogen labeled N15 isotopic form. Into it, Escherichia coli was added. Escherichia coli was allowed to grow for many generations. Many generations. In 20 minutes completes one generation, 40 minutes 2 generations. So that way it was allowed to grow for quite few days. After few days, obviously it will have produced many generations. Then the solution was taken out. Okay, the solution, from the solution the E. coli were taken, and then they were subjected to centrifugation with cesium chloride. After centrifugation, the solution of cesium chloride showed a dense region where the E. coli had accumulated. When investigated, it was found that it contained the heavy DNA, that is N15. Heavy DNA means that E. coli, the DNA in the E. coli were having nitrogen. Which nitrogen? The isotopic form of nitrogen. As we all are familiar, DNA has nitrogen in the form of nitrogenous bases. So the new Escherichia coli that were developing, in those Escherichia coli, the N15 form of nitrogen incorporated. They took from the source and they started growing. So all the new Escherichia coli, the DNA of all the new Escherichia coli were labeled with N15 forms. So here we have that original N15 DNA. Here the DNA has been shown, DNA of the Escherichia coli. Next, again, from this particular solution itself, okay, mind it, not new Escherichia coli. From here itself, which had already grown for many generations, from there few E. coli were picked. Okay, some batch of a little bit of E. coli were picked up, and then they were placed into another solution which contained N14 forms, which contained N14 form of ammonium chloride as the source of nitrogen. This time it was allowed to grow for just one generation and centrifuged, and they were centrifuged. After centrifugation, only one fragment was obtained even this time, but this fragment was found to be less dense, less heavy than the previous case. So it is indicated in this manner, little raised, the position is given, and when its DNA was tested, its DNA was found to be containing both the type of nitrogen, N15 as well as N14. Immediately after one generation, this was taken out. So generation 1 in N14 medium showed that the DNA in Escherichia coli, each of the Escherichia coli, not separately, each of the Escherichia coli had both N15 and N14 form of nitrogen. So it was concluded that one, now where from will N15 come? Because we are providing what? N14. So N14 should have entered into the E. coli in order to produce new DNA copies. all should have been N14, but it was seen even N15 is continuing, even N15 is present just after one generation. From where did this N15 come? Obviously from the previous generation. So it was concluded that the new DNA that developed, definitely in them, 50% of it was parental DNA with that N15 form, and the new one that was formed, it must have been from the source, new source N14. So it was almost confirmed in that manner that probably the new DNAs had 50% of the parent type and 50% was new type. One more generation was allowed to grow here. After one generation, little bit was taken up. The rest was left for one more generation. That means after 40 minutes. The first case is after 20 minutes. The second is after 40 minutes. After 40 minutes, when E. coli were taken out and then they were subjected to centrifugation, this time it was found that two fragments of E. coli. One fragment, when investigated, was found to have hybrid DNA like after the first generation, and the second fragment, which was found to be lighter than the other one, had only N14 forms. It indicates generation 2. It indicates that the source of nitrogen is N14. It's continuing. So the first generation offsprings now will behave as the parent for the second generation. So these are behaving as the parental strands. These parental strands, what happened? They are hybrid. So during formation, what happened? They separated, and because N15 is present here, so N15 continues. It does not break down. The DNA does not break down. It continues, but it took up a new strand, the complementary strand. It took the materials from the ones that were available from the source. It took up. It also produced a pure N14. How did it do? The complementary one was from the source that was available, N14. So N14, it is behaving as the parent and it is getting N14 from the source. So what happened? It turned out to be pure N14. But the other one again became a hybrid. Same thing for the other strand. If we continue for a third generation, what will happen? The number of pure ones will go on increasing. Pure means N14 ones will go on increasing. But the N15 will not die out. N15 also continues. It was same. So through this experiment, Michelson and Stahl, they proved that during DNA replication, the parental strand is conserved. It does not break down, but it is not fully conserved. That means the whole of it does not remain. What happens in one DNA? What happens? It gets separated and it takes up a complementary strand from the source, from the new available source. So here again the same experiment, just to make it more clear. So medium containing N15 heavy isotope, the Escherichia coli were grown, which is considered as F0 generation, the first sample. Okay, where heavy isotope will incorporate into the genome. So here, then they were allowed to grow in N14 forms, where after 20 minutes the F1 generation showed hybrids. After 40 minutes, they showed hybrid as well as with N14. And after 60 minutes, as I told you, third generation, what is happening? The number of hybrids decreasing, but the number of N14 increasing. Why? Because we are providing the source of N14. It is taking from the surrounding medium. Then if we continue, the number of the hybrid forms will go on decreasing, but it does not disappear completely. Conclusion: DNA replicates in a semi-conservative manner. Now we come to the process of DNA replication. What is DNA replication? It is the copying mechanism of DNA. It was proposed by Watson and Crick in 1953, as I already mentioned. the two strands of DNA separate during the formation, each one will act as a template, as the parental strand for the synthesis of new complementary strand. That means the two strands of DNA, the first step they will separate out, and then they will take from the environment new nucleotides and form a polynucleotide. A polynucleotide new one will be formed, which will form hydrogen bonds with the single parental strand. So what will happen? Two parental strands will form two complementary strands new. So we get two complete DNAs. It results in synthesis of a DNA molecule with one parental and one newly synthesized strand, hence called as semi-conservative. Now, what are the things required for DNA replication? The main components required: DNA-dependent DNA polymerase, that is the enzyme. It is the main enzyme required for DNA replication. Next enzyme required is DNA ligase and primase helicase. So these are the three main types of enzymes required for DNA replication. Other than that, deoxyribonucleoside triphosphates. This is a compound which will act as a substrate. Also, it will act as an energy molecule to provide energy for this polymerization. Polymerization reactions require a high level of energy that will be provided by this triphosphate molecule, this deoxyribonucleoside triphosphate. One RNA primer is required, and obviously the parental DNA strand. So these are the main components required for DNA replication to take place. Now we will learn the procedure. Process of DNA replication. So what happens? First, this is the DNA strand. This is the if we just observe this part, okay, it is a DNA strand, the blue ones. That means it is a DNA strand 5 to 3, 3 to 5. The first step is with the help of the helicase. What happens? The hydrogen bonds are broken, followed by movement of DNA polymerase, and that will go to form the replication fork, from where there is a point called as origin of replication. So DNA, the polymerase, the other enzymes, they will investigate. Okay, they will move throughout the DNA, investigating, and wherever they find the origin of replication. Now, what is origin of replication? It is made up of some specific bases at which the enzyme will get attached. Also, we can say these are the active sites of enzymes. So, what will happen? They will bind upon that, and at that point, breaking of hydrogen bonds starts. Separation of the two strands will start. There can be quite a number of origin of replication along a DNA. So, at different points, DNA replication can take place simultaneously. The polymerase catalyzes. DNA polymerase is an important point to remember, can catalyze polymerization only in the direction of 5 to 3. It can catalyze only in the direction of 5 to 3. So what happens after the helicase and polymerase opens up the DNA? At this point, the DNA has been opened up. It forms a replication fork. This replication fork that has been formed now, it will expose the parental strands. The bases of the parental strands are exposed. As you can see here, this blue, they are exposed. When exposed, now what will happen? The whole event is happening either in the nucleus in case of eukaryotes or in the cytoplasm in case of prokaryotes. So both of the environment, they will have nucleosides, deoxyribonucleoside triphosphates abundantly with different bases. Some will be with adenine, some will be with thymine, some will be with guanine, and some will be with cytosine. They will be present. So accordingly, if suppose here is a nucleotide with thymine base, then a trinucleoside with adenine will come and bind up. If here there is one with cytosine, then one nucleoside with guanine will come and form a hydrogen bond. So it will go on proceeding in this manner. Proceeding of the formation of the new strand will always take place in 5 to 3 direction. Now 5 to 3 dispolarities we have already discussed, we have already talked about in a previous lecture. Okay, we have already talked about, so I'll not go into those details. So it is assumed that the students, they understand what is the meaning of 5 to 3. So the formation will always be in the direction of 5 to 3 by rule. Naturally, it takes place in that direction. And also we know that the two strands of DNA have to be in opposite directions, and the polarities have to be opposite. So what happens now? These two, the first strand, the parental strand, which is 3 to 5, will start with replication first, because immediately as the direction is 5 to 3 new formation, so opening up is taking place in the same direction, so it goes on forming. As the parental strand is opening up, new bases go on adding. Furthermore, if it opens up here, it can continue forming. So that strand will be called as leading strand. That strand will be called as leading strand, and this formation is known as continuous formation. But complications will arise with the other one. The other parental strand is with the polarity 5 to 3. Now formation can take place in 5 to 3. Formation can also take place in 5 to 3. Now 5 to 3 formation is not possible like the previous one. Why is it not possible? Because if 5 to 3 in this direction formation takes place, both parental and the daughter strand will have the same polarities, which is not possible. It is just impossible. It cannot happen. Polarity of the other one has to be 3 to 5. It has to be 3 to 5 because the first parental is 5 to 3. But formation can take place in 5 to 3. So what will happen? Whatever portion of parental DNA has opened up, that portion, for that portion, the new DNA will be formed, starting from the opposite end. Starting from the opposite end, but following the rule 5 to 3. So a small stretch of DNA is formed first, whatever portion opened. Next, another portion opens up, then what will happen? It will form a second fragment, second part of DNA. Again, the third one, it will form one more fragment of DNA. So, as the DNA opens, it forms in the opposite direction. That is why what happens here, breaks occur. Because when this much is open, okay, fine. The entire length, new complementary one has been formed. But it cannot proceed here. Formation was in the opposite. As this DNA will open again, what will happen? New one will form from the opposite direction again. As it opens, again from the opposite direction. So consequently, the new DNA that will be formed, the complementary strand that is formed with the taking the template 5 to 3, there it will be discontinuous. The formation will be discontinuous and it will be slower even compared to the first one. So it is called as lagging strand. And the smaller fragments developed will be called as Okazaki fragments. Later, DNA ligase, another enzyme. Later, the DNA ligase will come and or will play the role here by joining together all of the fragments. All of these individual fragments will be joined together. The Okazaki fragments will be joined together, will be glued up together with the help of the DNA ligase enzyme. So, cells divide for growth and for continuation of species. First step in cell division is doubling of its genetic material, which occurs during S phase of cell cycle, and it is DNA replication. Okay, it controls the functioning of life. Replication of the DNA is the mechanism that doubles the DNA content in cells before they actually divide, that is during interphase. S phase DNA replication happens. It helps in equal distribution of DNA in the new daughter cells. Okay, so that is all for DNA replication for now, and next day I'll move on with a new lecture that is on transcription.